than 60% of the diet. So keep that in mind when you're comparing the animal and the human studies.
Thank you.
My name is Tony Sclafani, and I study rat sugar preference and appetite. I do not study addiction and won't comment on addiction. But concerning the provocative statement by Dr. Lustig about fructose, we've published 30 papers, at least in rats, and every time we give them the choice between glucose and fructose, they prefer glucose. Glucose is a much more rewarding sugar to rats than fructose. I just wanted to make that clear.
Do you give them sucrose?
We give them sucrose. If you give them sucrose versus maltose, they prefer maltose because it gives them twice as much glucose. Glucose has a very potent reinforcing effect when you equate for taste. That's true for the rat, it's true for the mouse. I don't know about humans.
Thank you.
My name is Amy Lin from Worcester Center for Carbohydrate Research at Purdue University. The question came from Dr. Lustig, one slide, but this is a question for all speakers and maybe to some audience. You mentioned sucrose. We have an enzyme to hydrolyze it, to hydrolyze the alpha-1 to linkage. This enzyme is sucrase. So this enzyme actually locates in our small intestine and is anchored on the membrane surface. And sucrase is on the terminal anchored on the membrane. So it's related to the enzyme reaction in the small intestine. So since it's enzyme reaction, if we have a lot of products, it may slow down the reaction or inhibit it. So I'm very curious, when we consume a big bottle of like a soft drink, we consume a lot of sucrose. How much time does it take to make a sucrose equal to the mixture of glucose and fructose in our body?
Well, I'll take a stab at this one. I think there's a lot of data. There's data from several labs that would shed some light on this. If you look at serum glucose concentrations, for example, feeding high fructose corn syrup in comparison with sucrose, you see that the time that they both appear in the bloodstream in the same time course. They're superimposable. And so I think that says that there's not a significant delay during the hydrolysis step.
I do not disagree with you, but you mentioned blood sugar. We found actually blood sugar may not be the best method to examine the glucose released from a carbohydrate source. Maybe if you can use carbon-13 to label it, and then you can test it in a more precise way. But again, I do not have data. I do not disagree with you, but just a comment here. Great. Thanks.
Thank you very much. 2 more questions.
Richard Black, Kraft Foods, First Penny. Congratulations on no fear under fire here with the crowd. And the presentation is really very good. I just want to make a plea on talking about addiction. I've seen so many times a slide put up that says, wow, there are the parts of the brain that light up with cocaine addiction, and oh, sugar hits the same system. It's the flip. We're supposed to respond to food, to good tasting things. Those areas are supposed to light up with those parts of our behavior that make a difference. It just so happens that cocaine also hits those areas inappropriately. And so if we just remember that it's not any food hitting our addiction centers or our cocaine centers. It's, yeah, there may be some people, I have no idea, I'm not an expert in this area, that overreact and underreact to stimulants in those centers. But it's cocaine that's the weird thing, not food.
Thank you very much.
Wait, before you leave, I have a question for you. Sir, I have a question for you. You have children?
Yes.
I have 3.
Good. How many times did you have to introduce a savory food to them when they were babies before they'd accept it?
I'm not typical. Our family's not typical.
10 to 13 by Leanne Birch's data.
Well, so let me say our family's not typical.
Okay. How many times do you have to introduce?
Our kids eat all kinds of food. We never actually had food eating problems except for green vegetables with the oldest kid.
How many times did you have to introduce a sweet food before the child would accept it?
Appropriately enough. Most all infants, it's been shown, react positively to sweet taste from birth because breast milk is sweet.
Breast milk is not sweet.
Yes, it is.
Got
And they better
lactose
respond
has a sweetness
positively
index
to
of
breast
15.
milk.
Come
They
on.
better respond positively to breast milk, otherwise they won't eat it. It's an appropriate response.
I want to just say to Richard Black that we did some work for Kraft Nabisco, and the only time my kids were impressed was when I came back from Kraft with a bag full of every kind of Oreo ever made. So they thought I was a pretty famous guy at that point.
That's pretty good.
Thank you. Last question.
Hi there, John Stephen Piper from McMaster in Hamilton and University of Toronto. I want to thank all the speakers for really excellent and engaging talks, and I think it's important that we do have a debate on this. I think as part of that debate, though, we have to be moving away from looking at these ecological analyses and observational analyses where we don't really have adequate control. And even some of the animal data where we've heard that really the applicability is an issue. I mean, even with regards to de novo lipogenesis, we see in the rat and mouse that you can push it with high carbohydrate feeding to 50 to 90%, whereas in humans it's very hard to push past 10%. And as we saw when it's looked at carefully by Luke Tappy, you're looking at probably less than 3%. So I think it's really important we move away from these, and I think guidelines have done that. I mean, the highest level of evidence we have are controlled feeding trials and nutrition. That's what's cited as the highest level of evidence for the dietary guidelines, as well as for clinical practice guidelines. And that was really the reason we did that with our knowledge syntheses and meta-analyses. We wanted to take the highest level of evidence and then synthesize that. And when we did show, and I thank the speakers for showing it, that under isocaloric conditions, across conditions, lipids, glycemia, actually where we saw a benefit, uric acid, some of the data that I'd given to Rob, he showed, but I haven't actually published yet on NAFL and others, we don't see a signal for a detrimental effect, whereas under hypercaloric conditions, right across the board we do. And we look at that question carefully, using formal tests for interaction, we see it's actually the energy and not the substrate that seems to be driving the signal that we see there. So I think that's important. Now to Rob Lustig's very, very important criticism, and Jim Rippey mentioned the same thing, we're looking just at pure fructose. These were not high fructose corn syrup or sucrose. But what I would say is we did look at sucrose and high fructose corn syrup as the comparator. About a third of the isocaloric trials had it as a comparator. And when it was the comparator, fructose behaved no differently than starch or high fructose corn syrup or sucrose or lactose or any of the other carbohydrate comparators. So, and I think part of the reason for that is the fructose, even though it may be given alone in some of the acute studies, in long-term studies, when you design a trial, you're giving it in a mixed meal context. So you're giving it with a meal or you're giving it with other foods where you would have rapidly available carbohydrate, which then would provide the glucose load for them to be taken together. So I think that those are important considerations. The only other point I would make is regarding fruit. I think that there's a real problem. People, I think we need to speak a lot about fruit and the benefits of fruit, because I have an endocrinologist, and this is a teaching hospital at the University of Toronto, which was telling their patients not to consume fruit based on a lot of these data. And I think that that's a really dangerous road to go down. And when we look at certainly at the level of fructose that is obtainable from fruit, we see a benefit for glycemic control, no adverse effects across the board. And when we actually look at fruit in our studies, our glycemic index studies, we see that it's actually the most important determinant of the low glycemic index and relating to our effects on A1C and HDL and others. So I would just add those comments.
Could I just say, John, if you look at de novo lipogenesis, many animals can turn a lot of carbohydrate into fat.
Exactly, 60 to 90%.
Yeah, it's just humans that can't do it.
They can't, exactly. Yeah, that's what I'm saying. I agree.
John, your meta-analyses were absolutely fascinating, and I debated including some of them. The problem I had was that you were picking fructose replacement for mostly starch in your studies, which eliminated a lot of studies and came down to a smaller group of individuals. And we don't put much fructose in our food supply as fructose. And the point you were making is essentially all with glucose, even in the beverages where the fructose is a significant amount. And so I had trouble putting that into the context of these other studies, for example, the Marsk study where they fed or where they provided beverages as a 16-ounce or two 16-ounce beverages for a period of time, and where they use liquid, because you put it in the way most of yours did, it's as a solid.
Actually,
and solids are very different from
about
liquid.
47% were in fluid form.
Yeah. But they're very different responses between the two of them. So I had that problem with it. And maybe you can go back, because you're such a smart guy, and pull some of those things apart from me, because I'm too dumb to do that.
Well, what I would say is, no, and thank you for that comment, and Russ D'Souza, a postdoc at Harvard that I know is working with you, he's actually looking at that question. So we're now looking at fructose-containing sugar-sweetened beverages. So our fructose alone, sucrose, high fructose corn syrup, bring them all together so we can look at and explore some of those subgroup effects to see, is fructose really different than sucrose or high fructose corn syrup across all the studies?
That'll be very interesting.
Great. Well, thank you all for hanging in there, and please thank our speakers for their excellent talk.